Hello everyone, and welcome to another tutorial for BioSE1A. This tutorial is about understanding DNA replication. This is a DNA replication fork. On the diagram, label the circled ends as to whether they are 5' or 3'. Before we answer this, let's quickly review DNA replication. There are 3 steps for DNA replication. Step 1, unzipping, unwinding, and strand separation. Step 2, adding complementary bases. Step 3, formation of 2 identical DNA molecules. Let's look at these steps in more details. First, initiator protein binds to double-stranded DNA at the origin of replication, and using ATP energy, slightly unwind the DNA. Then DNA helicase binds to the unwound DNA and continues unwinding and breaking the hydrogen bonds between base pairs. As the strands separate, replication forks form. Single-stranded binding proteins stabilize the unwound parental DNA. Then DNA polymerase forms the new DNA molecule by traveling along each single strand and adding complementary nucleotides. Then DNA polymerase can only add nucleotides to the 3' end of an existing nucleotide, so it relies on primase to synthesize a short RNA primer. The leading strand is synthesized continuously in the 5' to 3' direction. The leading strand requires only one priming. The lagging strand, however, is synthesized discontinuously and requires a series of RNA primers. Primase synthesizes a short RNA primer, which is extended by DNA polymerase to form an Okazaki fragment. The RNA primer is then replaced by DNA by another DNA polymerase. DNA ligase then joins the Okazaki fragment to the growing strand. Finally, 2 identical DNA molecules are produced, each consisting of one new strand. Now back to the question. Label the circled ends as to whether they are 5' or 3'. To do so, first can you identify which is the leading strand and which is the lagging strand? The lagging strand has the Okazaki fragments. Knowing that the leading strand is synthesized continuously in the 5' to 3' direction, then this is the 3' end. And this is the 5' end on the leading strand. Once you identify the ends of one strand, the rest is easy. Since the 2 strands are antiparallel, this makes this end the 3' end, and here's the 5' end on the leading strand template. Now you can identify the ends of the lagging strand template. Here's the 3' end, and the other end is the 5' end. Then the lagging strand, which is antiparallel to the lagging strand template, will have the 3' end on this end, and the 5' on the other end. Next, in which direction, left or right, is the replication fork proceeding? Since the direction of the unwinding of the parent strand is to the left, the replication fork is proceeding to the left. Now clearly indicate where helicase will be found and what is its function. Remember, DNA helicase unwinds the double helix, so ahead of it, the DNA strands are still connected, while behind it, they have separated. So the helicase is found at the edge of the double helix. Next, circle the location of the nucleotide that was most recently added. In the leading strand, it is synthesized continuously in the 5' to 3' direction. So the nucleotide with the 3' end is the most recently added, which happens to be the nucleotide closest to the replication fork. For the lagging strand, it is synthesized discontinuously, forming Okazaki fragments. The most recent nucleotide added is in the Okazaki fragment closest to the replication fork. But remember, DNA polymerase still adds to the 3' end. So the most recent nucleotide is the nucleotide with the 3' end on the Okazaki fragment closest to the replication fork. How many DNA polymerase enzymes would you expect to find at the replication fork? You would expect 2 DNA polymerases, one on the leading strand and one on the lagging strand. What enzyme is responsible for stitching the short fragments on the top strand together? DNA ligase joins the Okazaki fragment to the growing strand. Why can't DNA polymerase perform this function? Remember, DNA polymerase can only add new nucleotides to a pre-existing 3' OH group. DNA polymerase requires the energy released from hydrolysis of DNTP in the form of the incoming new nucleotides to drive phosphodiester bond formation. So DNA polymerase cannot form the final phosphodiester linkage to stitch together Okazaki fragments because no new nucleotides are added. Instead, DNA ligase performs this function. On which strand is primase required more often, and why is it necessary? Since DNA polymerase cannot initiate DNA synthesis without having a primer to start with, the lagging strand is synthesized discontinuously and requires a series of RNA primers. That concludes this tutorial. Thank you for listening, and good luck.